Diversity of microbes, fungi, and protists. Figure 13.1. Living things are very diverse, from simple single-celled bacteria to complex multicellular organisms. Chapter Outline. Section 13.1. Prokaryotic Diversity. Section 13.2. Eukaryotic Origins. Section 13.3. Protists. And Section 13.4. Fungi. Introduction. Until the late 20th century, scientists most commonly grouped living things into 5 kingdoms: animals, plants, fungi, protists, and bacteria, based on several criteria such as absence or presence of a nucleus and other membrane-bound organelles, absence or presence of cell walls, multicellularity, and mode of nutrition. In the late 20th century, the pioneering work of Carl Woese and others compared nucleotide sequences of small subunit ribosomal RNA, which resulted in a dramatically different way to group organisms on Earth. Based on differences in the structure of cell membranes and in rRNA, Woese and his colleagues proposed that all life on Earth evolved along 3 lineages called domains. The 3 domains are called bacteria, archaea, and eukarya. 2 of the 3 domains, bacteria and archaea, are prokaryotic, meaning that they lack both a nucleus and true membrane-bound organelles. However, they are now considered, on the basis of membrane structure and rRNA, ribosomal RNA, to be as different from each other as they are from the 3rd domain, the eukarya. Protists were the first inhabitants on Earth, perhaps appearing approximately 3.9 billion years ago. Today, they are ubiquitous, inhabiting the harshest environments on the planet, from boiling hot springs to permanently frozen environments in Antarctica, as well as more benign environments such as compost heaps, soils, ocean waters, and the guts of animals, including humans. The eukarya include the familiar kingdoms of animals, plants, and fungi. They also include a diverse group of kingdoms formerly grouped together as protists. Welcome back to Concepts of Biology. We'll continue to read Chapter 13, Section 13.1, Prokaryotic Diversity. By the end of this video, you will be able to describe the evolutionary history of prokaryotes, describe the basic structure of a typical prokaryote, identify bacterial diseases that caused historically important plagues and epidemics, and describe the uses of prokaryotes in food processing and bioremediation. Prokaryotes are present everywhere. They cover every imaginable surface where there is sufficient moisture, and they live on and inside of other living things. There are more prokaryotes inside and on the exterior of the human body than there are human cells in the body. Some prokaryotes thrive in environments that are inhospitable for most other living things. Prokaryotes recycle nutrients, essential substances such as carbon and nitrogen, and they drive the evolution of new ecosystems, some of which are natural while others are man-made. Prokaryotes have been on Earth since long before multicellular life appeared. Prokaryotic diversity. The advent of DNA sequencing provided immense insight into the relationships and origins of prokaryotes that were not possible using traditional methods of classification. A major insight identified 2 groups of prokaryotes that were found to be as different from each other as they were from eukaryotes. This recognition of prokaryotic diversity forced a new understanding of the classification of all life and brought us closer to understanding the fundamental relationships of all living things, including ourselves. Hi! If you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. Alright, let's get back to reading. Early life on Earth. When and where did life begin? What were the conditions on Earth while life began? Prokaryotes were the first forms of life on Earth, and they existed for billions of years before plants and animals appeared. Earth is about 4.54 billion years old. This estimate is based on evidence from the dating of meteorite material, since surface rocks on Earth are not as old as Earth itself. Most rocks available on Earth have undergone geological changes that make them younger than Earth itself. Some meteorites are made of the original material in the solar disk that formed the objects of the solar system, and they have not been altered by the processes that altered rocks on Earth. Thus, the age of meteorites is a good indicator of the age of the formation of Earth. The original estimate of 4.54 billion years was obtained by Claire Patterson in 1956. His meticulous work has since been corroborated by ages determined from other sources, all of which point to an Earth age of about 4.54 billion years. Early Earth had a very different atmosphere than it does today. Evidence indicates that during the first 2 billion years of Earth's existence, the atmosphere was anoxic, meaning that there was no oxygen. Therefore, only those organisms that can grow without oxygen, anaerobic organisms, were able to live. Organisms that convert solar energy into chemical energy are called phototrophs. Phototrophic organisms that require an organic source of carbon appeared within 1 billion years of the formation of Earth. Then, cyanobacteria, also known as blue-green algae, evolved from these simple phototrophs 1 billion years later. Cyanobacteria are able to use carbon dioxide as a source of carbon. Cyanobacteria began the oxygenation of the atmosphere. The increase in oxygen concentration allowed the evolution of other life forms. Figure 13.2. This hot spring in Yellowstone National Park flows toward the foreground. Cyanobacteria in the spring are green, and as water flows down the heat gradient, the intensity of the color increases because cell density increases. The water is cooler at the edges of the stream than in the center, causing the edges to appear greener. Before the atmosphere became oxygenated, the planet was subjected to strong radiation. Thus, the first organisms would have flourished where they were more protected, such as in ocean depths or beneath the surface of Earth. At this time, too, strong volcanic activity was common on Earth, so it is likely that these first organisms, the first prokaryotes, were adapted to very high temperatures. These are not the typical temperature environments in which most life flourishes today. Thus, we can conclude that the first organisms that appeared on Earth likely were able to withstand harsh conditions. Microbial mats may represent the earliest forms of life on Earth, and there is fossil evidence of their presence starting about 3.5 billion years ago. A microbial mat is a large biofilm, a multi-layered sheet of prokaryotes, including mostly bacteria, but also archaea. Microbial mats are a few centimeters thick, and they typically grow on moist surfaces. Their various types of prokaryotes carry out different metabolic pathways, and for this reason, they reflect various colors. Prokaryotes in a microbial mat are held together by a gummy-like substance that they secrete. The first microbial mats likely obtained their energy from hydrothermal vents. A hydrothermal vent is a fissure in Earth's surface that releases geothermally heated water. With the evolution of photosynthesis about 3 billion years ago, some prokaryotes in microbial mats came to use a more widely available energy source, sunlight, whereas others were still dependent on chemicals from hydrothermal vents for food. Figure 13.3. This microbial mat grows over a hydrothermal vent in the Pacific Ocean. Chimneys, such as the one indicated by the arrow, allow gases to escape. This photo shows stromatolites that are nearly 1.5 billion years old, found in Glacier National Park, Montana. Fossilized microbial mats represent the earliest record of life on Earth. A stromatolite is a sedimentary structure formed when minerals are precipitated from water by prokaryotes in a microbial mat. Stromatolites form layered rocks made of carbonate or silicate. Although most stromatolites are artifacts from the past, there are places on Earth where stromatolites are still forming. For example, living stromatolites have been found in the Anza-Borrego Desert State Park in San Diego County, California. Some prokaryotes are able to thrive and grow under conditions that would kill a plant or animal. Bacteria and archaea that grow under extreme conditions are called extremophiles, meaning lovers of extremes. Extremophiles have been found in extreme environments of all kinds, including the depths of the oceans, hot springs, the Arctic and the Antarctic, very dry places, deep inside Earth, harsh chemical environments, and high radiation environments. Extremophiles give us a better understanding of prokaryotic diversity and open up the possibility of the discovery of new therapeutic drugs or industrial applications. They have also opened up the possibility of finding life in other places in the solar system, which have harsher environments than those typically found on Earth. Many of these extremophiles cannot survive in moderate environments. Now on to biofilms.